Okay. Guys, I don't know. I don't know what's going on. I don't know why it's like I don't know why it's like glitched. Um all right. Oh my god. Okay. All right. I think we're good now. Um the lo-fi for some reason was glitching out. Um let me see. Well, obviously the stream restarted. Uh for some odd reason. Okay. All right. I don't know why the stream just like it just crashed. That's like never happened before. That's like incredibly strange. Um, but yeah. All right. So now we have that. Okay, so basically what I said before the stream crashed, the videos no longer up, so I'll explain it again. Basically, uh I was saying that this is gonna be the last day that I do this is gonna be the last day that I use the Captain Hook deck. If I see that I'm taking like more than like an hour and a half on the deck, there's no reason why I should be taking that long. So I'll probably end up switching to On King, and then I basically will call it a day. Um, like obviously, I have a lot more cards done in PanCal, so it's not too late to make the switch, and I'll probably just switch into the Captain Hook deck for Psych Soch because I have all of my PanCal progress in this deck. So yeah, that'll be pretty much what I do. Um, but in the meantime, before I start, let me get my charger because I forgot to get it. I forgot to grab it before, and obviously we might be here for a while. All right, so um, oh, okay, all right, that should be good. All right, let's get it. Um, we're gonna zoom through these reviews, or not, maybe not, but um, we're gonna try to zoom through these reviews. All right, how does one calculate isoelectric point? Isoelectric point is going to be the average between the for a neutral amino acid. It's going to be the average between um, between the carboxyl group and the amino group. Um, in an acidic one, it's going to be the acid group and the carboxylic acid group, the acidic R group and the carboxyl group. Um, in The basic ones is gonna be the basic one averaged with the amino group. Damn. Do I genuinely have this down like that? I don't know if this is accurate. So guys, I may have messed some stuff up. I'll be honest with you. I think I think I need to adjust this. I think I had it set up a certain way, and I optimized the preset. Um, Hold on, bro. I don't want, bro. I do not want 60 cards, bro. Do not give me the 60 cards yet. It's training to regulate your self attention, meditation. Okay, that's more like normal. With the other FSRS settings, this shit would have been like 2 months. Okay, I don't know why it said 2 months. Like I don't think I like. Okay, I guess maybe I kind of did know it that well, to be honest. But I wouldn't give myself that much rest time. Um, to hear sound, you need pressurized sound waves and hair cells. Electron affinity is the it's like the electronegativity is the force, right? Is the force used, and electron affinity is like I don't know, like the amount of this amount of energy, right? The relative amount of energy used to pull an electron. Oh man, energy released when it gains an electron. That was like it was like on the same line, but it wasn't quite the answer. How much mass? 1/2000th of a proton. In order of vibration, the oscilles include the we have the malleus to the incus to the stapes. Um, this is gonna be what kind of proteins? Proteins destined for secretion and integration into the membrane are located at the front. Reduced memory, judgment, and concentration. Nice. With internal, no, because it can be symmetrical. Caffeine, the hammer. The manifest content, the amount of energy needed to remove an electron. Oh man, I really want to memorize that gaseous atom part. What is a cofactor coenzyme? Um, this is going to be a non-protein molecule that binds to non-protein molecule that binds to an enzyme to help provide it with its function. Something like that. Opioid effect is gonna decrease CNS activity, decrease not brain pressure, blood pressure, heart rate, act as an analgesic and vasodilate. Middle ear is the region with the three ossicles. Hallucinogens. They inhibit adenosine receptor and can disrupt sleep. Okay, so we have, um, where is it? Oh, right here. Okay, so we have, dear King Philip came over from Great Spain. So we have domain, kingdom, phylum, class, order, family, genus, species. Now that was smooth. Okay. Form. Psychoactive can alter consciousness and perception. It can act as a stimulant, hallucinogen, or depressant. Cilia in the ear are called kinocilium. No, hair bundles. The basilar and tectorial. The shared structure of flagella and cilia in eukaryotic cells. It's the 9+2 structure with the 9 doublets on the exterior and the 2 central doublets of microtubules. What is viral extrusion? It's when a virus um, it's when a virus exocytoses from the cell instead of killing it. The common depressant drugs are alcohol, barbiturates, and benzodiazepines. Delta waves are associated with deep sleep and comas. Light from the left side of the visual field, it hits the left nasal and the right temporal retina. Sensory adaptation. Treatments for insomnia include psychological training, medication, and lifestyle changes. Uh, Benzodiazepines, because there's no axons in the way of light entering the cell. Stimulants. Um, Stimulant, depressant, hallucinogens, and opiates/opiates will gain or lose electrons in a way so as to receive a noble gas configuration. Indications of sleep apnea include snoring and morning fatigue after a full night's rest. Is the neurotransformation process. Narcolepsy. Purpose of the oval window is to it's to push fluid through the cochlea. Shape theory of olfaction or the stare theory. Damn, I had this inversed, but I still feel like that's more. I feel like that's right. Like, there's no other shape theory. So, lag phase is when the bacteria are getting adapted to their environment. Smell synapses at the orbital frontal cortex. Sound waves that pass through the auditory canal will hit the tympanic membrane of the eardrum. Protein associated taste tint. No, glutathione. Um, Decreased CNS activity, decreased blood pressure, decreased heart rate. One more. Oh, decreased brain processing speed or processing speed. And then it vasodilates at low concentration and vasoconstricts at high ones. People that undergo hypnotism are typically um, people that undergo hypnotism are typically more susceptible to suggestion and exhibit alpha waves. Eardrums will cause the ossicles, pheromones, sensory neural, narrow sensory neural hearing loss. Damn, I had it the right way. I don't know if I should give myself that. I'll be honest with you. I feel like I got it, and the test is multiple choice. I'm gonna give myself it. How does one calculate isoelectric point? Um, didn't I? What? Didn't I just do this? I just did this. Oh, that was before. Oh, so it didn't even change any. It didn't really change much. If this was the normal one, it didn't change a lot. It just put it further. Maybe I should change it back then. Um, this is C fibers. What is the log phase? The log phase is um that's when the bacteria have adapted to their environmental conditions, and so they're like starting to their population is growing. C spindles are bursts of brain activity, 90 minutes. GPCRs, stimulus. Okay, you see, that's how I know that I still have the things sped up. We've heard this song like every day. What is the function of connective tissue? Um, what is the function of connective tissue? Um, it's gonna support it's it's gonna support the organs. It's gonna secrete. Um, okay, I know that the last one is that it's gonna provide some of the materials that are necessary for the ECM. But what is the second one? It's something, right? Is it gonna be something upon which the parenchyma depends? Like provide a framework for epithelial cells to carry out their function. Damn. What is electron affinity? It is the energy released when an electron gains an when an atom gains an electron. Wait, I just clicked again. I had that. What is the nucleus? The nucleus is the control center of the cell, surrounded by a double membrane. Um, it houses genetic information and other machinery to transcribe DNA into RNA. Parvocellular relies on cones, right? Yeah, cones. What do rows? The energy level of the valence electrons. What is ionization energy? Um, it is the amount of energy necessary to remove an electron from a gaseous atom. Uh, bar, mm, yeah, barbiturates. Should have just gone with my instinct. The inner ear includes the semicircular canals and cochlea. Methadone is used. It's used to fend off addiction. Um, when it's dark, rod cells are turned on. This is gonna activate. If it's gonna inactivate if they're turned on. It's gonna inactivate the on sensors. Um, Psychoactives, circadian rhythm. The lytic cycle is going to enter the cell, overtake the machinery, um, assemble its machine, assemble its own proteins, and basically like produce itself, and then it's going to lyse the cell and kill it so that it can spread to other cells. It doesn't integrate into the genome. What is the Golgi apparatus's function? The Golgi apparatus is going to function to um, what's it called? The Golgi apparatus is going to be. I'm like searching my head for like the picture of the card. Um, it's going to be a series of stacked sacks of membrane of stacked membrane bound sacks that package, modify, and sort proteins for transport. Damn. I think I was given the definition for what is Golgi apparatus. Damn. Cribriform plate separates the olfactory epithelium from the brain. What is a peptide? A chain of amino acids. The stationary phase is when they are running out of resources, and so like their population growth like staggers out. What is the function of lysosomes? To digest material like foreign materials or damaged slash unneeded organelles. To be converted into a neural impulse. Um, Accessory olfactory bulbs during N1. Pain and temperature. 20 hertz and 20 kilohertz. Uncompetitive inhibition. Um, Uncompetitive inhibition is oh, what is uncompetitive inhibition? Uncompetitive inhibition basically just means that It means that the inhibitor is going to it's going to bind to the enzyme substrate complex, and it's going to lock it in. So it's going to bind to the enzyme substrate complex and lock it in to prevent it from releasing any product. That was like really hard. What are the two types of chemical bonds? Um, Covalent and I feel like I must be missing something here. I'm gonna say I'm gonna say I'm gonna say covalent and ionic, but that's because that's an answer that I remember, but it's not really the answer that I would give. Yeah, I don't know why it says that. I guess they're called intermolecular forces, but I don't know. I feel like I learned it as a type of bond, so it's kind of hard. What is viral lysis? It's when the viral basically enters into the cell, it takes over the machinery, and then it just lyses the cell to spread uh, its little its children viruses. What is the restriction on bacterial conjugation? F plus can only conjugate to F minus, not to other F plus though. There are more rods than cones. Amplification. What is Vesper theory? It's a it's a means of predicting the shape of a molecule based on its bonded and unbonded electrons in the Lewis structure. No, temporal region don't they don't cross over. It's a chain of more than 20 amino acids. Yeah, I think. What are the general characteristics of metalloids? There are no specific characteristics or general characteristics. It's just sometimes they might have more in metallic characteristics or more in non-metallic characteristics. They might have some of each. Steps to olfaction include so the molecule binds to the olfactory epithelium cell, then which is going to release a G protein. The G protein is going to open ion channels. The ion channels are going to are going to lead to an action potential that is going to go up to the sensory glomerulus or the olfactory glomerulus, and then go to the myelotucted cell, and then go, and that action potential is going to synapse in the brain. Okay, I didn't say that it was going to go through the cribriform plate, but I knew that. It needs to go through the cribriform plate. What is the function of connective tissue? So it's gonna support the organs. It's going to provide the framework for epithelial cells to function, and it's going to provide the some of the materials that are necessary for the ECM. Mesopic. How does a non-competitive binds equally to the free enzyme and to the enzyme substrate complex? So it's going to decrease the Vmax, and the Km is going to stay the same. I think it binds to the allosteric site. So, and since Vmax is staying the same, it's going to be like, oh, it's going to be like, bro, it's going to be like this. Like something like that. Not like that exactly. More like this line should be more something like this. Yeah. Is when a subject gives a negative response to a present signal, a miss, group one, ion channels, between the basal body. Oh, it connects the basal body to the filament. Um, Olfaction, barbiturates, induce sleep and reduce anxiety. Memories that um, incorporate the hypnotizer's expectations of an induced state of consciousness. It's a series of interconnected membranes that um, that are contiguous with the nuclear envelope. Histidine structure is boom, 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 boom. We're gonna go N, N, and both of these are gonna point toward the corner. Okay. Opiates slash opiates. What is a ligase? It's gonna join. It's gonna catalyze addition slash synthesis reactions between large molecules. The most commonly known one is in nucleic acid synthesis. What is the lysogenic cycle? Um, the lysogenic cycle is uh, it's when the virus enters the cell and it integrates itself into the genome so that it can reproduce its own like parts. As the host cell reproduces, the virus obviously reproduces with it. Um, and yeah, what is the Golgi apparatus's function? It's gonna package, transport, package, sort, modify, package, modify, sort um, proteins for transport. Those modifications basically will assign a destination for the proteins, like in or inside or outside the cell or to the ER, etc. This is the energy released when an atom gains an electron. Glycine just has an H, gly, G. What is the catalytic efficiency? It's going to be Kcat divided by Km. What is the nucleolus? That's going to be the It's like a dark spot, and it's a structure inside the nucleus that produces our RNA. What special case must you remember? If it has a full octet, it's going to be a lot harder to remove an electron. Light first hits the eye at the cornea. Theta. Stimulant or hallucinogen. They're brittle, non-lustrous. Uh, they can be gases. They're poor conductors of heat and electricity. Pineal gland. Theta waves, sleep spindles, and K complexes. All right, so we have um, caffeine, amphetamines, methamphetamines, cocaine, nicotine, MDMA. Stapes is attached to the oval window. They suppress cortical arousal to keep you asleep, and they aid in encoding declarative memories. Send sensory signals to the metrotufted cells. DNA or RNA. Uh, DNA or RNA, and then DNA or RNA, single-stranded, double-stranded, circular, linear. They have more control of their attention, which I clearly don't. The steps from light hitting a rod to a signal getting sent to the brain include uh, um, mm, okay, so the light's gonna hit the rod. Then the rod is gonna turn off. Um, then that's gonna activate the bipolar cell, then the retinal ganglion cell. Then it's gonna send that to the optic nerve, and the optic nerve is going to send the signal to the brain. What is ionization energy? It's the energy needed to remove an electron from a gaseous atom. Yeah, I think that's it. What is the function of the mitochondria's outer membrane? To serve as a selective barrier between the the inner mitochondria and the cytosol. Um, if it's a if it's a metal, it's going to become smaller. If it's a non-metal, it's going to become bigger. This is the olfactory bulb. Sleep apnea. the number of valence electrons. A protein with a carbohydrate-based prosthetic group. The iris is a muscle. It's going to increase the brain's response to GABA, induce sleep, and reduce anxiety. Manocellular pathways get at determining motion and depth. What is the stroma? The stroma is the functional part of the tissue. It's usually composed of epithelial. Oh, damn. The stroma is not the functional part. I don't know what I was thinking. Is the state of being awake but daydreaming? Stimulants are drugs that uh, increase blood pressure, increase brain activity, increase heart rate, and vasodilate. The area in the nostril responsible for olfaction is um, the olfactory epithelium. It's the inactive form of an enzyme that needs to be activated. They rely on GPCRs. Golgi apparatus is a series of interconnected sacs that uh, modify, package, modify, sort um, proteins for transport. Those modifications can those modifications I don't know if that's even part of this question. Those modifications can bring something to a can create a destination for a certain protein. Thank you. Damn. Proteins and lipids. I'm not giving myself that. What it 6.636 times 10 to the negative 34th jouleseconds. A protein with a nucleotide based prosthetic group. One sigma bond and two pi bonds. Opioids are um, opiates are synthetic. Opiates are natural. Hallucinogens are drugs that distort perceptions, dilate pupils, heighten sensations, increase connectedness, and cause mood swings. Yo, the first time I ever did uh, Pankow, it was like I didn't. I literally it took me so long to memorize that card, like so long. What are dipole-dipole interactions? Um, they're a more permanent attractive force. Um, that's caused. Well, it's an attractive force caused by permanent dipoles, um, due to like just differing partial charges, um, or opposite partial charges. TRPV1. What is the function of connective tissue? Um, it's a support. It supports the body. Uh, Provides a framework for epithelial tissue to function, and secretes molecules that make up the ECM. I kind of want to do that one more time. What is electron affinity? Electron affinity is the energy released in when an atom gains an electron. It packages, modifies, and sorts proteins and lipids for transport. Those modifications can basically direct a protein or lipid to a specific destination. The stroma is a support structure upon which the parenchyma depends. It's composed of connective tissue. What is the Golgi apparatus? It packages a series of stacked membrane-bound sacs that package, modify, and sort proteins and lipids for transport. The stroma is a support structure of an organ upon which the parenchyma depends. It's usually composed of connective tissue. What is the function of connective tissue? Um, mm, um, damn. I just get like brain fog sometimes. And to think I slept well today. Like yesterday, I might have been a little sleep deprived, but today, I feel like I actually slept well. So there's no excuse today. What is the function of connective tissue? It supports the body. It provides a framework for epithelial cells to function, and it secretes various molecules that make up the ECM. Okay, that was like painful. Okay. Um. All right. Yet another day. I don't know, bro. I really want to set this to 60, but I don't know if we're gonna make it to 60. I'm gonna be honest with you guys. 60 sounds like it might actually take me until 3 in the morning. Um, I hope not, but I'm gonna stretch before we do this. Okay. All right. Well, what is the death phase? The death phase is when the bacteria have run out of resources and they start dying. I'm literally gonna suspend this card. Actually, no. I'll just do easy. What is the cytoskeleton? It's the structure within the cell that holds the cell together. Is there a separate question that is going to ask me what are the types of cytoskeleton? Is that a question? Okay, I don't think so. So I'm just gonna. What is the cell wall? It is that's like a barrier that only realistically mainly plant cells have um on the outside of their cell membrane. Okay, yeah. Well, my answer was wrong, but yeah. Rigid structure that forms well, partially wrong. That forms the outer barrier of the cell. What is a cell envelope? Okay, let me edit this answer. Okay. What is a cytoskeleton? The cytoskeleton is the structure within the cell that helps to provide support. Uh, they can serve as like transport pathways. Um, there are three of these, three of the main types of like cytoskeleton parts. It's like microtubules, microfilaments, and intermediate filaments. It's it's the rigid barrier. I don't know. What was it? What was it? Rigid barrier that forms the outer rigid structure that forms the outer barrier of the cell. Rigid structure that forms the outer barrier of the cell. What is the cell envelope? It's all of the layers starting extending outwards, starting at the cell membrane. What is the basement membrane? Underlying layer of connective tissue to which epithelial cells are attached. What is the cytoskeleton? Um, so the cytoskeleton is um, it is the network of three structures that provide shape or structure in the cell. Um, these can serve as like transport pathways. The examples are microfilaments, microtubules, and intermediate filaments. Network of three structural components. Okay. What is a cell wall? The cell wall is the rigid barrier. Damn, bro. Rigid structure that forms the outer barrier of the cell. The basal body is the membrane made of connective tissue, um, which tightly encloses the. It's gonna tightly enclose Um, Not tightly enclosed. What am I talking about? Um, it's like it's basically like. Oh wait, this is a different question. I'm talking about the basement membrane. Oh my god. The basal body is the part of the flagella or cilia that basically holds it to the cell. Complex structure that anchors the flagellum to the membrane. Yeah. What is now again? Now what is the basement membrane? Um, it basically supports the epithelial cells, underlying layer of connective tissue to which the base underlying layer of tissue of connective tissue to which the epithelial cells are attached. Okay. What is the cytoskeleton? The cytoskeleton is the network of structural components that provides shape or structure to the cell. They can serve as transport pathways like microfilaments, microtubules, and intermediate filaments. Okay. What is the cell wall? The cell wall is cell wall. It's the rigid structure forming the outer barrier of a cell. Um, it's found in almost all prokaryotes and in some eukaryotes, like plants. Oh wow, that was actually correct. That I feel like the definition applies to so many things that it's like it's not even specific. What is the basal body? The basal body anchors the flagellum to the cell. It also provides the motor force to for the bacteria to move. I guess the cell anchors the flagellum, a structure that anchors the flagellum to the membrane. Okay. What is the basement membrane? It is the membrane that attaches uh, the layer of connective tissue that the epithelial cells bind to. Yo, vaguely that was right. Wow. Okay. Underlying layer of connective tissue to which epithelial cells are attached. What is the cell wall? Rigid structure that forms the outer barrier of the cell. The basal body, it anchors the flagellum to the membrane. Um, it also provides the motor force to activate the flagellum. The cell envelope is all the layers starting from the membrane. long, hollow, helical structure composed of flagellum. What is the basement membrane? Basement membrane is the layer of connective tissue to which the epithelial cells are attached. What is smoothie R? Um, Smoothie R is the part of the ER that has no proteins, no ribosomes attached. Um, its functions, I don't know. Okay, so lipid synthesis, detoxification, and packaging and transport. Lipid synthesis, detoxification, packaging, and transport. Okay. What is the rough ER? The rough ER is going to um, it has ribosomes attached. It's a part of the ER with the ribosomes attached, and it functions obviously for protein synthesis. Uh, and I don't know what else. What is the bacterial flagellum filament? Um, what? Oh, uh, it is a long hollow helical structure that's made of flagellum. The smooth ER. What's the definition again? It just says the functions. It doesn't even. It literally doesn't even say what it is. Okay. I'm just gonna change the question because it's not asking me. It's not giving me the answer for that. What are smoothie R's function? What are the smooth ER's functions? Okay. What is the cytoskeleton? The cytoskeleton is the network of structural components that support that help provide shape slash structure to the cell. It's the network of three structural components. Um, they can serve as transport pathways, and examples are intermediate filaments, microfilaments, and microtubules. What is the rough ER? Um, what is rough ER? Is this the same thing where it doesn't tell me what it is? Bro, it just doesn't tell me what it is. You see how much more appropriate that actually makes it, that actually like that definition or that like phrasing is. It's like it directly answers the question or something. It's crazy. Um, the bacterial flagellum filament is a long, hollow helical structure made of flagellum. What are the smoothie R's functions? Um, uh, a lipid synthesis, detoxification, and packaging and transport. What is a cell wall? A cell wall is the rigid structure that forms the outer barrier of the cell. What is the basal body? It is the basal body anchors. It's a structure that anchors the flagellum to the membrane. Um, it also provides the motor force to activate the flagellum. What is the cell envelope? It's all of uh, the cell envelope is the cell envelope is okay. It's all of the layers. The name for all of the layers starting from the cell membrane. What does rough ER do? The rough ER is going to take materials from the cytosol in order to synthesize proteins and secrete them into the ER lumen for them to be sent out, probably into the smooth ER to be sent to the Golgi. What is the basement membrane? The basement membrane is the structure. Um, it's the structure made of connective tissue to which the epithelial cells are attached. Oh my god, bro. What is parenchyma? The parenchyma is the functional part of the tissue. It's made of epithelial tissue. Functional part of an organ. What is exocytosis? It's leaving the cell, basically, by like it like pushes the membrane out and it like pops. It like pops out like a bubble. Takes material into what is chemotaxis? Ability of a cell to detect and move to slash away from chemical stimuli. Um, what is the basal membrane? It is the underlying layer of connective tissue to which epithelial cells are attached. It is the functional part of an organ. It's composed of epithelial tissue. Basically, There are some molecules that are inside of a secretory vesicle, and it basically goes and fuses with the membrane in order to release the contents. What is binary fission? It's literally like it's exactly what it says. It's just the cell splitting. Chemotaxis is the ability of a cell to detect chemical stimulants. What is bacterial transformation? When a bacterium picks up foreign genetic material and integrates it into its own genome. Foreign genetic material and integrates it into its own genome. Okay. What are smoothie R's function? The smoothie R is responsible for lipid synthesis, detoxification, and packaging and transport. What is binary fission? It's a simple form of asexual reproduction seen in bacteria. It basically copies its own DNA and splits. The ability of a cell uh, to detect chemical stimuli. Transduction. Hold up, bro. Uh, I see. Okay. What is bacterial transformation? Um, Picks up foreign genetic material and integrates it into its own genome. The ability of a cell to detect and move away or to chemical stimulants or chemical stimuli. What does the Ruffy R do? The Ruffy R is going to take materials from the cytosol and it's going to use it in order to create proteins and then release the protein into the lumen to be released into the smooth ER. And then from there, probably be released to the Goji. What is bacterial conjugation? Um, okay, this is what I wanted to see like a long time ago. When bacterium forms conjugation bridge with another bacterium, I honestly like if I don't do badly today, I genuinely might like I might genuinely reorganize the cards. Like before starting, like we might just take some time and reorganize the order of the cards so that I can actually like learn like while I'm doing this. Like even that way, even if the use of Hassan videos aren't enough, like at least I've number one seen the cards because I was rearranging them, and in order to rearrange them, obviously I need to know where every card needs to go. Um, and it should make my life easier. It might take maybe like 20 minutes, but better those 20 minutes than 3 hours of time. Um, when bacterium forms conjugation bridge, another bacterium shares genetic information. Um, okay, so it has the F plasmid. Obviously, when it has the F plasmid, it gets the sex pilus, and bruh, it literally just copies the plasmid over there and is like, all right, you got it now too. That's funny. One bacterium forms conjugation bridge with another bacterium and shares genetic information. What is bacterial transduction? It's when a virus accidentally traps another bacteria's DNA and shares it with another bacterium. Well, I was mostly right, but not right enough. You see, this is the problem with these decks. Like, because how are you going to tell me that I really have 44 new cards and 15 of them I still haven't gone through? I've only released one card from the cycle. What is a cytoskeleton? The cytoskeleton is the network of three structural components that provide shape slash structure to the cell. Um, they can serve as pass like transport pathways. Like the examples are microtubules, microfilaments, and intermediate filaments. I think I said that, right? I think I said that. What is bacterial transformation? It's when a bacteria integrates foreign genetic material into its own genome. Uh, what is the bacterial flagellum filament? Uh, it's the it anchors the flagella to the membrane. It also provides the motor force necessary to activate the flagellum. Oh man, damn! I think I just that was its function. My bad. That was actually the basal body. Shaking my head, bro. The underlying structure, underlying layer of connective tissue to which the epithelial cells are attached. What is the basal body? The basal body, it provides the motor force necessary for the flagellum to activate the flagellum. Um, the basal body is it provides an anchor for the flagellum to the membrane. What is the cell wall? It is the layer that rigid structure that provides the outer layer of the cell. Oh my god. Rigid barrier. What is parenchyma? The functional part of an organ. It's composed of epithelial cells. What is the cell envelope? Um, what is the cell envelope? Um, what is the cell envelope? Everything extending outward and starting from the cell membrane, like all the layers. Cell bacterial transduction is when a virus like takes in bacterial DNA and integrates it into another bacterium's genome. What is the bacterial flagellum filament? Um, it is a long, hollow helical structure composed of flagellum. It is the ability of a cell to detect and move away from or to a chemical stimulant. I think that's correct. What is exocytosis? Um, basically, a secretory vesicle has some molecule. It goes up to the cell membrane. It fuses with it and releases its contents. Apoptosis. It's like cell death, programmed cell death. Okay. What is an obligate anaerobe? Yes, I love these cards. Give me more. Give me all of them actually. Obligate anaerobe, obligate aerobe. Give me all of them, bro. Obligate anaerobe. It means that it must use anaerobic reproduction, not reproduction, respiration, anaerobic respiration. Um, uh, uh, mm, yeah, I think it just has it. It can't use it cannot use oxygen, and oxygen is basically like toxic. Like it dies in the presence of oxygen. Needs a non-oxygen environment to survive. That's kind of what I said at the end there, but it just it didn't really mention that it needed to use anaerobic respiration. What is binary fission? Um, it's like simple asexual reproduction. Apoptosis is programmed cell death. Bacterial conjugation is um, okay. I don't remember the terms of it. When the bacterium forms a conjugation bridge with another bacterium. Okay. What are the smoothie R's functions? It's going to be responsible for lipid synthesis, uh, detoxification, and packaging and transport. What is an obligate aerobe? Um, I mean, by that logic, it should die in the presence of not oxygen. Yeah, yeah, right, kind of. An episome. Uh, is that like the plasmid? Yeah, plasmid that's able to integrate into the host bacterial genome. What is an obligate anaerobe? It means that the bacterium dies in the presence of oxygen. What is an endosome? Endocytic endocytic vesicle that pinches off from the membrane during endocytosis. Conjugation, it's when a bacterium forms a conjugation bridge with another bacterium and shares genetic information. See? Yeah. Bacterial transformation. Um, it means that the bacterium takes in genetic material, like foreign genetic material, and integrates it into its own genome. What does Ruffy R do? It's going to take materials from the exterior, from the cytosol, and it's going to use it for proteins, release it into the lumen of the ER, and then that ER, that like molecule is going to butt off in a vesicle to the smooth ER. It's going to travel to the smooth ER to then be packaged and transported to the Golgi apparatus. What is an episome? It is a plasmid that can be integrated into the host bacteria to the genome. Yeah. What is an anaerobe? It's a bacteria that doesn't use oxygen to survive. What is an endosome? An endosome is an endocytic vesicle that basically enters the cell, like that enters the cell by butting off of the membrane going inside, like yeah, that pinches off from the membrane during endocytosis. What is an aerotolerant anaerobe? An aerotolerant anaerobe is an anaerobe that can use either anaerobic or aerobic respiration, so it won't die in the presence of oxygen. Um, oh, damn. So I don't know. I don't. Maybe it's facultative anaerobe that can use it. Can use both. What is a virulence factor? A plasmid making bacteria more pathogenic. Okay. What is a transposon capable of inserting and removing themselves? Okay. What is a tail sheath? The long, thin part of a bacteriophage. What is an anaerobe? A bacteria that doesn't need oxygen to survive. What is bacterial flagellum filament? It is the long, hollow, helical structure made of flagellum. That was kind of hard. What is an endosome? An endosome is an endocytic vesicle that pinches off from the membrane during endocytosis. What is a sex pilus? The sex pilus is what helps to form the conjugation bridge during conjugation. Appendages that male bacteria use to form conjugation bridges. What is a virulence factor? Virulence factor is a plasmid that make bacteriums more pathogenic. Okay. What is an aerotolerant anaerobe? An anaerobe that doesn't need oxygen to survive. It doesn't use oxygen in its metabolism, but it can survive with oxygen in the environment. Um, what is a transposon? It is genetic elements that can insert or remove themselves from the genome. What is a tail sheath? It can act kind of as a syringe. It's like the long, thin part of the virus. The long, thin part of the virus that is see, that's why I'm saying I'm actually having fun now. The long, thin part of the virus that um it's the long, thin part of the virus which can act as a syringe. Obviously, I said like of a bacteriophage specifically. Um, can act as a syringe to inject its genetic material. What is a sex pilus? It's the appendage used by male bacteria to form conjugation bridges. What is a cell wall? It is the rigid layer that forms the outside barrier of um, outside layer of the cell. What is parenchyma? Parenchyma is a functional part of an organ. It's comprised of epithelial tissue. What is the basement membrane? The basement membrane is the it is the ground basement membrane. It is the underlying layer of connective tissue to which epithelial cells are attached. Boom. What is an obligate aerobe? Um, it's a bacteria that needs oxygen to survive. What is a sex factor? Um, it's the plasmid in male bacteria that basically makes for the sex pilus. Yeah. What is bacterial transduction? It's when a virus captures bacterial bacteria genetic information, in order to transfer it to other bacteriums and like into the genome of other bacteriums. What is bacterial conjugation? Uh, it's when like a bacterium forms a conjugation bridge and transfers the F plasmid over. What is a retrovirus? What is a sex factor? The sex factor is basically the plasmid um, that allows for the formation of the sex pilus with which they can do conjugation. What is chemotaxis? The ability of a cell to detect and move away from or to chemical stimulants. What is a provirus or prophage? It's like the offspring virus. Oh, there are different things. Provirus is viral DNA integrated into a eukaryotic. Um, What is bacterial conjugation? Okay. What is bacterial conjugation? Halfway through, kind of. More like a quarter way through, but halfway through. Um, what is bacterial conjugation? Bacterial conjugation is when they form, like bacteria form a sex pilus, or well, they have a sex pilus, and they form conjugation bridges to share genetic information. What is a retrovirus? It's an enveloped single-stranded RNA virus that has a reverse transcriptase. Um, so when it enters the when it enters the cell, when it enters the whole cell, it can transform. It can synthesize DNA from its single-stranded RNA and integrate itself into the genome. I just feel like it's so like it's such like a roundabout mechanism. Like, okay, so you have you're trying to tell me that you're an RNA virus, and instead of just already being DNA, you want to be RNA. Like, I don't know. That sounds pretty amateur to me. Like, not to say like like it's not valid, but I just feel like that's an attack on the virus itself. Like, it's actually ass. Like, HIV obviously shouldn't exist, but I'm just saying it should die for real for just trying, bro. Because at this point, enveloped single-stranded RNA viruses that carry an enzyme called reverse transcriptase. Um, enveloped single-stranded RNA viruses that carry an enzyme called reverse transcriptase. I pretty much said this. I'm gonna give myself a good. What is a valence factor? Valence factor is a plasmid that makes a bacterium more pathogenic. What is a transposon? Genetic elements that can remove or insert themselves from the genome. Apoptosis is programmed cell death. Bacterial transformation is when the um, bacteria intake like genetic information and integrate it, like foreign genetic information, and integrate it into their own genome. What is a provirus? A provirus integrates itself a uh, into a human's genome. A prophage integrates itself into a bacteria genome. Oh, eukaryotic cells genome. I don't know. I just read the word human before, and I was just like, "What is a tail sheath?" Um, It is the long thin part of a bacteriophage that can act as a syringe in order to inject genetic material. Obligate anaerobes, they will die in the presence of oxygen. What is a sex pilus? It is the appendage used by male bacteria in order to form conjugation bridges. What is an obligate aerobe? They need oxygen to survive. What is an episome? It is a plasmid that's capable of integrating into the host bacterium's genome. What is the bacterial flagellum filament? Uh, it is a long, hollow, helical structure. Um, I don't remember the last part. Composed of flagellum. Damn. Okay. Okay. What is an aerotolerant anaerobe? It won't die in the presence of oxygen, but it doesn't use it in its metabolism either. What is an anaerobe? That it doesn't need oxygen to survive. Endosome is an endocytic vesicle that pinches off of the membrane. Yeah. What is a sex factor? It is the plasmid that allows bacteriums to form sex policies with which they can perform conjugation. Sex pili. Provirus or prophage. Provirus is like a virus's genetic information that integrates into a eukaryotic cell, and the other one is into a bacterial cell. What is the bacterial flagellum filament made of? Oh no, not made of, but bacterial flagellum filament is a long helical structure that's composed of flagellum. What is a plasmid? It's like a circular, like additional region of DNA. Small pieces of circular DNA acquired from the environment. What is an endosome? Um, it's an endocytic vesicle that's taken in during endocytosis. Endocytic vesicle that pinches off from the membrane during endocytosis. What is a peroxisome? Not what it does. Okay, that's kind of hard. I don't know. It's like a vesicle that uh, it breaks stuff down. It's a vesicle that breaks stuff down. That's all I know. Single membrane bound organelle containing peroxide. What is a mitochondrion? Um, it's the powerhouse of the cell. What? Double membrane organelle with structures in its inner membrane that allow it to generate ATP. Ugh, that's such a boring explanation. It's the powerhouse of the cell, bro. What is a plasmid? Um, Plasmid is small pieces of circular DNA. That are usually obtained acquired from the environment. They don't contain critical genetic information. That's it. What is a peroxisome? Um, it's a I think did it say it was single layered? Um, it's like a single layered organelle, uh, membrane bound organelle that contains peroxide. A single membrane bound organelle. Oh, yeah, it is. It did say single membrane. Okay. What is a lumen? A lumen is like the inside of a given organelle. Yeah, I give myself easy on that. What is a mitochondrion? It is a structure with it's a double membrane. It's a structure with a double membrane, um, whose inner membrane allows it to generate ATP. What is a histone? A histone is like my guess is it's this thing that DNA wraps around, um, and it kind of helps to suppress certain parts of the DNA so that they're not like being transcribed. It also is used for like compression, um, or compactness, when it comes to forming like the chromosomes during mitosis and stuff. Or did I mean mitosis? I don't know, bro. We're not there yet, bro. Just chill on me, bro. Protein and nucleus that eukaryotic DNA winds around to organize it. What is bacterial conjugation? Bacterial conjugation is um, basically they form conjugation. Bacteriums are going to form conjugation bridges to share genetic information. What is a peroxisome? It's a single membrane bound organelle that contains peroxide. What are flagella? Flagella are um, it's these like movie things, like they move in like circles. It's like a propeller. Structures involved in cell motility by spinning and flailing, generating force for movement. Um, the filament is a long, hollow helical structure comprised of flagellum. What is a virulence factor? It is a plasmid that makes a bacterium more pathogenic. What is a histone? Um, it is a structure around which DNA wraps itself to organize itself. Eukaryotes and archaea use them. It's a protein in the nucleus. Okay. What is an endosome? Um, hold up. Let me get my other. Uh, what is an endosome? It's an endocytic vesicle that um, it's an endocytic vesicle. I should find like a game to play when I start to lose attention, like you know how people be doing like the Pomodoro thing, like I feel like I would do the Pomodoro thing if I could find something that that would be still kind of related, but I don't know, that would make me want to that would give me like a mini goal like along the way. Um what is an endosome? Yeah, endocytic vesicle that pinches off from the membrane during endocytosis. What is a retrovirus? A retrovirus is an enveloped single-stranded RNA virus uh, that has a reverse transcriptase in order to synthesize DNA from its RNA and integrate itself into the host cell. I meant the genome, obviously. What are flagella? Flagella are structures involved in the motility of the bacterial or of the cells. Um, they accomplish that by spinning and flailing to generate force for movement. Wow, word for word. What is transposon? Genetic elements that can remove or insert themselves from the genome. What is a sex pilus? It is the appendage used by male bacteria to form conjugation bridges. What is a histone? It is a structure found in eukaryotic DNA to organize itself. It's used by eukaryotes and archaea. A protein in the nucleus. Why can't I say that? What is a plasmid? It's circular DNA in bacteria that contain non-critical genetic information. Um, they might using uh, those plasmids might offer them some kind of evolutionary advantage. But what is a mitochondrion? A mitochondrion is a structure. It's a structure whose inner membrane. It's a double membrane bound structure whose inner membrane has structures that allow it to generate ATP. I said structure, but like it's like organelle. Like I feel like that's good enough for me, bro. What is a tail sheath? A tail sheath is the long, thin part of a bacteriophage that can be used that can act as a syringe to inject genetic information into its host. An obligate aerobe needs oxygen to survive. Provirus is um, viral DNA that integrates into like a eukaryotic genome. And prophage is bacteriophage DNA that integrates into a bacterium genome. What is a sex factor? Sex factor is the sex factor. It's the plasmid that allows the bacteria to form to perform to bro to form a sex pilus and form conjugation bridges. What is a histone? It is a protein in the nucleus around which eukaryotic DNA wraps around to organize, and eukaryotic cells and eukaryotic organisms and archaea use them. What is a facultative anaerobe? This one uses both. It can use either one. What are cilia? Um, Projections from a cell involved in movement along cell surface. What is a centrosome? This is just about the DNA region where centrioles are found. Oh, I I definitely confused it with centromere. I'm not gonna lie. Centrosome is um where centrioles are found. What is a centriole? Uh, it's like, isn't that like two microtubules together? Oh, a hollow structure composed of nine triplets of microtubules found in the centrosome. Okay, yeah, I'm gonna need the definition for the centrosome again. What is a capsid? It's the protein structure around the virus, protein coat in the virus. Yes. What are cilia? Um, it's a structure involved in movement of, I don't know, like materials on the surface of the cell. Projections from a cell involved in movement of material along the cell surface. What is a bacteriophage? It's a virus of bacteria. Yes. What is a centrosome? Centrosome is the region where centrioles are found. Oh, that was I thought I needed to see it again, but maybe not. What happens if a light? Yo, am I bugging? What the? Yo, I think I'm being trolled, bro. What happens if a lysosome releases? I'm not crazy, guys. No, I promise. I'm not crazy. What happens if a lysosome releases its enzymes? Um, if it releases its enzymes, bro, it would probably break down the whole cell. It probably lyse. Oh, yeah. Okay, nice. I didn't I didn't expect that to be completely right, bro. I thought it was gonna be some other complex ass answer. I didn't know it was called autolysis, but I got it mostly right. So I'm gonna give myself a hard. Um, what is a centriole? Uh, a centriole is it's like a structure made of nine triplets. What is a peroxisome? A peroxisome is a single membrane-bound organelle that contains peroxide. What do we mean when we say epithelial cells are polarized? I don't know. One side of the cell attached to underlying structural cells, other is exposed to lumen. What does it mean to be stratified epithelium? It means like there's multiple layers of epithelial cells. And my grandma knew that. What is squamous epithelium? It means the cells are like square, flat, are flat scale like epithelial cells. Is the is the surface of your skin scale like? Is that squamous epithelium? Where is squamous epithelium found? Air sacs of the lung. Oh, it's stratified squamous epithelium. So that was close. It's just not. It's not simple, but flat scale like epithelial cells. What is a centriole? A centriole is nine triplets of microtubules found in the centrosome. A hollow structure. I need to say that hollow structure part. What does it mean when we say epithelial cells are polarized? It means that one side is exposed to like the connective tissue and the other side is exposed to like the uh is it the outer environment? The lumen of organ or the outside world? Okay. What is an endosome? Is an endocytic vesicle that pinches off from endocytosis. Um, what is the bacterial flagellum filament? It is a long, hollow helical structure composed of flagellum. What does it mean to be simple epithelium? It means that uh, I don't know, bro. Single layer. What does it mean to be squamous epithelium? Um, it means that it's like flat, scale-like epithelial cells. What is a centriole? A centriole is a hollow structure composed of nine triplets of microtubules. What does it mean when we say epithelial cells are polarized? Um, what does it mean when we say epithelial cells are polarized? Um, it means it means that one side is exposed to the connective tissue and the other side is exposed to the lumen. Okay. What does it mean when we say, yeah, didn't I just say it? To lumen or the outside world? Didn't I say that? What are cilia? They're projections from the cell surface that allow for movement of materials um, along that very cell surface, obviously. Um, what is a capsid? It's the protein coat of a virus. Means it has one layer. What is a centrosome? Uh, it's the region where centrioles are found. You see, because why do you have so many like different like terms? Centriole, centrosome, centromere. Like, damn, bro. I understand it's a prefix, but goddamn, you didn't have to spam, bro. What are flagella? Flagella are um, I don't remember the definition for this. Okay. What happens if a lysosome release? It causes autolysis. The cell breaks down from the inside. What is a plasmid? Um, a plasmid is they're pieces of small circular DNA that do not contain critical information but might provide like an evolutionary advantage. What is a histone? Proteins in the nucleus around which DNA wraps it, like winds itself to organize. Um, like eukaryotic DNA is the one that wraps itself around there. But it's also used in archaea. What is mitochondrion? It is a double membrane bound organelle um, that has structures in the inner membrane that allow it to generate ATP. What are flagella? They're structures involved in cell motility, uh, accomplished by spinning and flailing, a lot like creating the force necessary for movement. Pseudostratified means that it looks like it's multiple layers, but it's not. What does it mean to be cuboidal epithelium? They're shaped. Now they're not shaped like squares, but they're shaped like cubes. Oh wow, I was right. What does it mean to be columnar? They're like really long cells, long, thin column-shaped cells. Yeah. What does it mean to be a prokaryotic cell? It means you have no nucleus, like a nucleus area. You don't have any membrane-bound organelles. What else? I think that's it. Oh, they have no nuclei, but they don't have membrane-bound organelles either. What is a facultative anaerobe? A facultative anaerobe. Um, it can exist. No, it can use both. It can use both. Yep. What is a bacteriophage? A bacteriophage can today today it's looking because we're doing so good. It's been 2 hours and 10 minutes. We're doing so good. I think today might be our first day of U World. I think we might do our first section of U World today. This might be our first U World passage. Um, we gonna work. We gonna do a lot today. Today's gonna be today's gonna be a good day. We gonna make sure of it. We'll make sure of it. What is a bacteriophage? It's a virus for bacteria. Pseudostratified. It looks like it's multiple layers, but in reality, they're all connected to the basement membrane. What do it mean to be squamous? They're like flat scale-like cells. Cuboidal, they're cube-shaped. Columnar, they're long, thin, flat cells. Long, thin column. Okay. What what is a centriole? It is a structure composed of nine triplets of microtubules. What does it mean to be? It has no nucleus. What does it mean to be a male bacterium to have a sex pilus or an F plasmid? What does it mean to be a female bacterium to have no F plasmid? What does it mean to be a eukaryotic cell? It means you have membrane-bound organelles and you have a nucleus. Yes. See, you see, because this one has membrane-bound organelles, but all the prokaryotic cells didn't. Okay. What does it mean that mitochondrion are semi-autonomous? Because it has its own DNA. Is that why it's saying? Contain some of their own genes, have their own ribosomes, can divide independently of the cell via binary fission. I didn't know that it could perform binary fission, but what does it mean to be columnar epithelium? It means that the cell, columnar epithelium are thin, long, column-shaped epithelial cells. What is a centriole? It's a it's a hollow structure composed of nine triplets of microtubules. It means that the bacteria is spiral shaped. What does it mean to be a female bacterium? Uh, it thinks it's going to take me 17 minutes. It means that it has no F plasmid, um, no F plasmid, and it accepts um, the genetic information from the male bacteria, and it obviously accepts the conjugation bridge. Wait, it says what? It has an F minus plasmid. Is that even true? Does it have an F minus plasmid, or does it just not have the plasmid? Because that was my understanding. Does bacteria the do female bacterium have an F minus plasmid? No, it does not have the F minus plasmid. It doesn't have the F. I don't know why it said that. It doesn't have an F minus plasmid. That's strange. Yeah, I give myself plus. What does what does it mean that mitochondrion they have their own DNA? They can split by themselves. They have their own ribosomes. What does it mean for a bacterium to be Kochi? It means it's circular shaped. Oh, close enough. But I'm gonna give myself again, but close enough. What are flagella? On the next one round, I'm gonna definitely click good. What are flagella? Um, flagella are um, they're projections that um, projections. Uh, they're structures that are involved in cell motility. Um, they accomplish this by spinning or flailing um, in order to generate force for movement. What does it mean for a bacterium to be Kochi? It means that it's sphere shaped. This means bacilli. Yikes. I don't remember. Shaped like rods. Okay. What color do gram positive and gram negative? It's like purple and Is it purple? Is it blue and pink, purple and pink, something like that? Purple and pink. Gram positive is purple. Oh, but they both start with a P. Damn. Negative. Negative pink and positive purple. Purple. Maybe I should call it purple. Positive purple. Positive purple and negative pink. You can't say positive punk. That don't make sense. But positive purple. What are the two types of single-stranded RNA viruses? I don't know. Positive sense and negative sense. Okay. I don't like how that ended there, but it's fine. I don't really want to continue doing this deck. I think we'll finish off the deck tomorrow and we'll maybe do it like 50-50. Like we'll do we'll do the 20, I think there's like 20 something cards left in this deck. So we'll do the remaining 20 and then we'll do the rest in Pankow. What does it mean for a bacterium to be bacilli? Um I think that's a good strategy. Like every time I just go straight to Pankow, like as a remainder, if I don't finish a deck, then it'll make it a lot easier for if I need to make the switch. Like today we had a very good day though. Like I think maybe it genuinely was that I needed to go to sleep. Like what does it mean for a bacterium to be bacilli? It means that it's rod shaped. It means it's one layer. The protein coat. What does it mean? One side is exposed to the structural tissue, aka the connective tissue, and the other side is exposed to lumina, the outside world. Positive purple, negative pink. What are cilia? Cilia are um, what's it called? They're projections from the cell. That allow for movement of materials along the cell surface. What is a centrosome? It's the area where centrioles are found. Positive sense and negative sense. What happens if a lysosome releases its enzymes? It causes autolysis. Squamous are flat, scale-like epithelial cells. It means you have no nucleus and you're a bum, pretty much. What does it mean to be a malobacterium? It means that you have an F plus plasmid and you can form conjugation bridges. Oh, I didn't even expect that to be actually part of it. It means that it looks like you have more than one layer, but in reality, all of the cells are connected to the basement membrane. It means that your cells are shaped like a cube. What does it mean to be a eukaryotic cell? It means that you have a nucleus and you're membrane bound. Oh damn! Why did I say why said eukaryotic cells are membrane bound, bro? I mean, they have membrane bound organelles. I'm gonna give myself a day on that. If I can remember it tomorrow, it's definitely not because I clicked that. To be honest with you, there must have been outside forces praying against me. To be honest, what does it mean to be columnar epithelium? Um, it means that uh, the cells are thin, flat. Um, um, the cells are thin, flat and column shaped. What is centriole? It is a microtubule. It's a it's a structure. It's a hollow structure made of 9 triplets of microtubules. It means that you do not have the F plus plasmid, but you accept the sex pilus from, or the conjugation bridge from the male bacterium. What does it mean that mitochondria are semi-autonomous? They have their own DNA, have their own ribosomes, and they can split on their own. It means that you're shaped like a sphere. Um, what are flagella? Uh, flagella are these are structures involved in cell motility, and they accomplish this by spinning or flailing, generating the force for movement. Means shaped like a rod. Two types of RNA viruses are positive sense and negative sense. Positive, purple. Okay. So okay, that means tomorrow we have 44 cards. I thought we had a lot. I thought we had a lot less, but guess I was wrong. Because we only did 20. We only did 20 yesterday, so that's why. All right, we can sync that. 123. We're on good timing. Probably we'll be going for another, like, if I had to guess, maybe like 20 minutes or so. Okay, so let me set up the like. So, hold up, let me see something. Okay. Um, okay. So, um, yeah, let me do close out this. All right. So, hopefully Jack Sparrow goes easy on me today. Um, and by easy on me, I mean I hope that even if he goes hard on me, I go hard too. You know what I'm saying? You feel me? All right. has like eight paragraphs, bro. You, Jesus Christ. I forgot to pause the lo-fi. That was like, I think that's it. I don't think I can give myself more time. If I'm staying true to the time, honestly, I'm even a little bit over right now. But okay, nice. Yeah, that's literally exactly what I was thinking. Like, what does the wheel have to do with literally anything? I could have sworn I like imagined like that for this question. Literally the only reason why I didn't pick this one is because I didn't remember like I like pictured while I was reading it someone is standing like in a cave like around like a fireplace, but I didn't know why I was picturing that. That was the only reason why I didn't get this question wrong. Yo, this one was scary. Nice. You did good today. Oh, bruh, bruh, bruh. I can't believe this. All of that. All of that is. I was like, wow. I actually did well. And the whole time they were all like, Yeah, all of the questions, literally all of the questions were easy, bro, except for one of them. Yo, I hate when this happens. This is like the second time that I'm like, oh, nice, I finally got all the questions correct, and it was an all easy passage, bro. This is incredible, incredible. All right, let me load up. Let me pull up your world. We're gonna new window. Okay, your world. Um, okay. All right. Let me look for my all right, and then we're gonna switch over again. All right. Well, let's do this. It's gonna be our last thing for the night. So it's just okay. I put it for 6 questions. Um, we'll do, which should basically be like one passage. We're gonna do it timed, not tutor. Uh, yeah, I think that's a chapter we literally just did today. So generate test. Highlight important text for fast reference. Next, 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 next. Which of the following molecules is most likely found within the cell membranes of intestinal epithelial cells? Um, Within the cell membrane, uh, cytoskeletal filaments. This is in the cell wall. Cholesterol might be. Definitely not cellulose. It's one of these two. Okay. Which will cause disease by inducing enterocyte lesions after it is grown in media with EHEC that contain S after plasmid. Which which process most likely? I would say conjugation. EHEC is classified. This type of classification is most likely based on the morphology. Which proteins identified in Figure 2 have the shortest amino acid sequence and the greatest concentration in WT cells, respectively? A small antibodies used to detect their epitopes with equal effectiveness. Um, have the shortest amino acid sequence. Uh, and the greatest concentration in WT cells. Shortest amino acid sequence and greatest concentration in WT cells. It's TIR and ESPB. Is there going to be a question that actually makes me read the passage, or based on the passage which best explains the Western blot results of type 3 secretion proteins in Lux S? Um, what best explains the Western blot results of type 3 secretion proteins? Yes. E. coli cells culture in BCM. I'll come back to that. Researchers are studying a single-cell pathogen. Which experimental method would be least effective for determining whether a pathogen is prokaryotic or eukaryotic? Would be least effective. Single-cell to fluorescent anti-mitochondrial antibody. Testing the pathogen for ribosome size. Assessing the pathogen for the presence of pyruvate. Um, well, uh, this one is kind of hard, to be honest. I still have 5 minutes left. Analyzing the cells for number of origins of replication. That might be not bad. To a fluorescent anti-mitochondrial. I think it's that one, bro. I think I have that right. Based on the passage, what best explains the Western blot results of type 3 secretion proteins in Lux S. E. coli cells cultured in PCM? Okay. Um, Based on the passage, what best explains the Western blot results of type 3 secretion cells cultured in PCM? Damn, bro. Well, I actually got that. I got the last one right and the first one wrong. What? Most people took 208 seconds. Yo, is this the question that they paused to read the question on? I'm sitting here taking both of these questions combined on this on number five because that's when I finally decided to read it. It was cholesterol. Damn. God damn. Damn, this really is in depth. Holy. The cell membrane is a phospholipid bilayer that separates the interior of the cell from the external environment. The two phospholipid layers are oriented such that the hydrophobic tails of the phospholipids in the inner and outer layers are in contact with each other, leaving the hydrophilic head groups in contact with either the intracellular or extracellular fluid environments. The cell membrane is described by the fluid mosaic model as a non-rigid phospholipid bilayer through which diverse proteins are scattered, the mosaic, and are able to migrate laterally. In addition to phospholipids, animal cell plasma membranes also contain cholesterol, glycolipids, glycoproteins, and other proteins. Cholesterol, which is found in eukaryotic but not prokaryotic cell membranes, functions to regulate the fluidity of the bilayer. Due to their unique structures, cholesterol molecules decrease fluidity at higher temperatures and increase fluidity at lower temperatures. As eukaryotic cells, intestinal epithelial cell membranes contain cholesterol. Choice A. All cells, eukaryotic and prokaryotic, have a plasma membrane, but some cell types are surrounded by an additional enveloping layer known as the cell wall. Unlike the plasma membrane, the cell wall is a rigid structure that defines the shape of the cell and provides added protection. Animal cells, including intestinal epithelial cells, do not have cell walls. However, nearly all prokaryotic cells have a cell wall. The major structural component of the bacterial cell wall is peptidoglycan, a polymer of sugars and short polypeptides. Yes, we'll probably be learning about peptidoglycans tomorrow. Um, you know what they say, positive purple. Um, okay. Cytoskeletal filaments are found in the cytoplasm of cells and can interact directly with various components of the cell membrane. However, they are not themselves part of the plasma membrane. Are you serious? I could have sworn that was part of the cytoskeletal that's like embedded in the membrane. Oh my god. Cellulose, a polysaccharide, is a primary component of the cell wall in plant cells. Cellulose is not found in the membranes of animal cells. Educational objective. The plasma membrane is a fluid lipid bilayer that contains a diverse array of proteins scattered throughout. Certain cell types have a cell wall surrounding their plasma membrane. However, animal cells do not have a cell wall. The primary component of bacterial cell walls is peptidoglycan. Well, this question literally like it didn't like genuinely like the entire passage didn't matter for this either. It's literally just asking why is it called the bacillus. This one you just had to read the figure. I feel like they love doing that. They just make you read the figure. But for this one I actually don't know why I got it wrong. I actually don't know how I got that right, so I need to read this. Oh, because wild type was positive lux, like it had the lux. TIR, what is TIR? Oh yeah, TIR was one of the secreted proteins. If I would have read this, I would have known. Like, I don't think I read that sentence. I think that's the reason why. Um, Now let's read. I don't even. I'm not gonna read the question. Read the questions like that. Okay, this one. This one was a little. This one wasn't too incredibly easy. Um, yeah, that's literally what I was thinking. Like the knot almost tripped me up, but like I figured, like in my head, it was like prokaryotes might have one origin of replication. Um, I didn't know anything about that anti-mitochondrial antibody. I wasn't sure what that would do. I wasn't sure if oh, but mitochondria have membrane. You see, that's the problem with memorizing prokaryotes as having like the definition didn't include that they don't have membrane bound organelles. You see, that's the flaw, the crucial flaw of Captain Hook. Shaking my head. Um, and I know that the subunits are different sizes. So okay, cool. We had a good day. We had a good day, bro. We got 83 on cars. We got 100 on this, like, or well, I guess not 100, but